Okay, so hey everyone, my name is Dharini Chokhadia, and I am the event manager and host for today's live podcast, The Next Experiment by the SciComm Synapse, a podcast featuring researchers and scientists. I am delighted to welcome our honorable guest and speaker, and we will be in conversation with Dr. Raghothanam M. Yennamalli on decoding life sciences with data. But before we begin, I'd like to introduce the SciComm Synapse. At SciComm Synapse, we strive to be a bridge between the world of research and the curious minds in our community. It's a space where ideas ignite, questions flourish, and discoveries inspire change. Guided by our beliefs in thinking, growing, and sharing, our mission is simple: to bring science closer to people in a way that's meaningful and inspiring. So, sir, I would like to, brief your introduction. Sir, shall I proceed?
Yeah, please.
Yeah, thank you. So, I welcome you all to another exciting episode of the SciComm Synapse, where we connect students and young researchers with inspiring scientists from diverse fields. It's a great pleasure to introduce our distinguished guest and speaker, Dr. Ragothanam M. Yennamalli, professor at the School of Computational and Integrative Sciences, Jawaharlal Nehru University, New Delhi. Dr. Yennamalli's research lies at the exciting intersection of biology, computation, and artificial intelligence. His lab leverages computational approaches to address complex biological questions, with applications in protein engineering, enzyme design, antimicrobial resistance, genomics, and sustainable biotechnology. In addition to his impactful research, Dr. Yennamalli is a passionate mentor and educator who has inspired many students to explore computational biology and interdisciplinary science. His career exemplifies how integrating biology with data science can drive innovation and solve real world challenges. So, today we will learn about his scientific journey, explore the evolving role of AI in biology, and gain valuable insights for students aspiring to pursue careers in research. Please join me in extending a very warm welcome to Dr. Ragothaman M. Yennamalli. So, sir, I would like to hand over the stage for you to present your slides. Thank you.
Okay. First of all, thank you to all of you for organizing this and giving an opportunity to talk about my work and also sharing questions and answering any queries from audiences. So I hope everyone's able to see my screen. So this is the only slide I have, and I'm probably going to take only like 5 minutes. So uh, the area of research I have been doing can be broadly divided into these 4 quadrants. And the first quadrant here is the first branch where I mostly deal with protein structures, enzymes, its dynamics, um, and then and the proteins that we had been working on with this virus targets, the dengue envelope protein that we started with, and now we are looking at enzymes. These are enzymes which break down carbohydrate molecules and chitin or any um, polymeric chains. So there are multiple uses. One is the direct one for biomass degradation for biofuel optimization. The other one is for human health, and that is something that we are working on right now. Then, as I said, the target branch we already started, and we are still doing something about using the dengue viral drug discovery and also the hepatitis C virus. We are doing some work in collaboration, and we are also looking at antimicrobial resistance and in terms of antimicrobial peptides that can be targeted. and there is a lot of work that we are doing in terms of a multi-target, antimicrobial peptide-based strategy. The third branch of work is in this lower right quadrant, where I have now been involved with genomics work and transcriptomics and integrative omics area of bovine genome. And we are looking at the native or indigenous cattle breeds in India, and we are looking at genetic signatures that are responsible for innate immunity in those bovine genomes. And also, we are looking at the comparative genomics work of a very interesting organism, Enococcus indicus DR1, which was isolated in India. And we are looking at the heavy metal tolerance and resistance that's present in this particular genome and the molecular mechanism behind it. So that actually has overlap with protein structure based work. Then the final one is about the most latest work that we have been working on in the last couple of years, which is transglutaminase 2 enzyme, which is involved in, or TG2 enzyme, which is involved in the celiac disease. And India has a population of about 1% who suffer from celiac, and there are also a lot of neurodegenerative diseases, which from time to time we have been doing some studies with phytochemicals and seeing their efficacy in how to mitigate the effects of these neurodegenerative diseases. Although we have been doing something, but my focus goes from time to time between one of these four branches. When it comes to the type of methodologies that we use to tackle all these four branches. We use structure-based bioinformatics, basically structural bioinformatics, where protein structures, its interactions, its the ligand interactions, and all those kind of techniques are used. We also use simulations, both fine-grained and coarse-grained simulations, to understand the biophysics behind these mechanisms, and also machine learning. And if there is, in the case of antimicrobial peptides and with drug discovery, we use virtual screen. So that's the basic overall of the work that has been happening at Yennamalli Lab for the last 15.
All right, sir. So it was a really great presentation of how the integrated biology and everything. So it is particularly real world presentation.
We
So
have we haven't gone yet to the translational aspect, but we are trying to, and hopefully in the next 2 years we'll have something with
yeah, sir. Just a
translation.
request that your voice is not properly
So
coming.
is it?
So
It's
yes,
not very clear. Is it very
yeah,
clear now?
yeah, yeah. It's good.
Okay.
It's
Okay,
yeah.
I've increased the volume. Sorry.
All
Yeah.
right. Yeah, yeah, no worries, no worries. So, thank you so much for the wonderful explanation. And now we shall proceed to the question section of our conversation to explore your insights in more depth. So the very
Sure.
first
Yeah.
question, yeah. So the very first question is every scientist has a story. Could you take us back to the beginning? Like what really inspired to pursue biology and how did you eventually find your passion into computational biology?
This is an interesting question. I come from a family of educators and artists. So there was no topics that were taboo while in the house. So we used to talk about many different topics while growing up, and at the time, my grandma, my grandfather, they would be very interested in the space exploration, the Apollo missions, and they would take us to the American consulate, and also we used to go to the British Council Library and so on. So there was a very healthy environment of talking about science. We used to have a lot of discussions on physics, on basically astronomy and many other things. But biology as such was more interesting because we had a huge garden and I was very interested in gardening. And then I would see that there were many caterpillars around that would go and infest these and then they will start, destroying the plant. So instead of removing all those caterpillars and saving the plant, what I would do is collect those caterpillars, put it uh, in a glass tumbler or a glass bottle covered with a mesh, and then feed the same leaves to them. And I want to see what they would turn up into. And they would most probably start building the stupas and then the chrysalis, which used to have this metallic sheen and it used to be very beautiful to look at and these amazing colors, and then they will metamorphose into either a moth or a butterfly. I have no idea. So sometime we used to like during the season when we get seasonal vegetables and the brinjal and the okra, or the lady's finger, they will have infestations. So I used to collect all these worms and then not have a fear or some kind of ick towards those things. That was one of the beginnings that I remember. But the major fuel that happened was during my ninth standard, when my biology teacher taught us biology in a very interesting way, and that pedagogical methods were extremely useful even now for me to when I teach my students. And her name is Kamalam Nayak, and there was a summer camp, and we went to Top Slip, which is a national park in the border of Kerala and Tamil Nadu. And it was a 3 day trip to that Top Slip area, Perambikulam National Park. And we saw a lot of these wild animals, we saw endangered species, we learned a lot about how to protect them, how environment plays a role, and so on. So she was the one who pushed me into getting into that summer camp and going to top slip and so on. And from then on, I had a very big interest in science in general, but most focused on biology. I used to read whatever I could lay my hands on biology textbooks and biology books that were uh, um, in our house. And also, I used to voraciously read, and that is something that I developed as a kid. We used to go to libraries, we used to read the World Book Encyclopedia, and then read. I used to read many things that were related to biology. And so when I finished my 12th, and then at the time I was trying for my medical, but unfortunately, I did not clear any of the exams. And I was looking at what to do next. And again, Kamalam Nair ma'am, she came into the picture and she said, do microbiology because of your interest. That will be a very good course for you and it will be something that you will like. And so I ended up doing my undergraduate in microbiology. And then I went to do a very good course, microbial gene technology from Madurai Kamala University. And at that time, I wanted to be in a university setup and I wanted to interact with the scholars, interact with scientists. And so I did not want to go to a college for my master's. I wanted to go to a university. So I had two choices, either go to a university outside the state or inside the state, and I picked up the one in Madurai. And Madurai was like at that time I didn't know, but after joining, I realized that is the cradle of biological sciences in India for a very long time. And Krishnasamy, not the latest one, but the one who started the School of Biological Science, had a vision. And at that time, the vision was completely different from other state university visions. And the only university which had a similar vision was MS University Baroda or JNU or University of Hyderabad. So NKU was a very different in that aspect, and I was very fortunate to have studied or to have learned the basics of genetics, molecular biology, and many other things from pioneers. And then I moved to JNU, for my diploma, and then I continued here for my PhD. And so that has been my journey into biology. And it was during the diploma I had a uh, like a dilemma, you can say. So I had option. I either can join NKU itself for the postgraduate diploma in bioinformatics, which was funded by the Department of Biotechnology, or the same program in JAIN. So which one should I opt for? Because it's the same course, the curriculum is the same, the funding agency is the same. There's not much difference. Only the cities or so that I thought. And but then my mentors they said that going to another university and especially going to the capital has an advantage, and I should take up the admission in JAIN. And so I ended up being at JNU. And the main motivation of joining bioinformatics, because until my masters I had done wet lab, so I was a trained biologist. And taking up bioinformatics was because we had seen these beautiful pictures of protein structures, that the how at the time we were just getting into bioinformatics, and bioinformatics was the hot potato in those days. I'm talking about 26 years ago. And so it was a very, very interesting, and we had visited the bioinformatics center at NKU and interacted with the scholars there. And they used to talk to us the research that they were doing. And I was very fascinated with the Markov chains, Markov models, and those kind of things. And so I thought, okay, rather than going into a wet lab biology, let me test how bioinformatics works, if that is to my liking. If not, I can always go back to the lab biology in my PhD. So that's how I ended up coming into bioinformatics.
Yeah, so that's a really, wonderful. But of course, there were a lot of challenges again. But we are happy that you were able to land up in the computational biology. So I believe that's an interesting perspective, and I think it naturally brings us to the next question. Many students are fascinated by computational biology, but don't know what it actually involves. How would you explain your research to a first year undergraduate in simple terms?
That's a good question, because when we were just learning the basics of computational biology or bioinformatics, we used to think that it's just a thing that you do on computer. More or less, that definition has still stuck around, and there is a inside joke and an ongoing academic joke that if you want to do bioinformatics, all you need is a laptop and good internet connection. That's all. It kind of loses the nuance there, but for an undergraduate person, bioinformatics is more than just sitting in front of a laptop and going through different databases, looking at the tools, downloading something, and getting some results. It is an interdisciplinary area, and you need to have some good foundations in physics, in chemistry, in biology, mathematics, statistics, and of course, in computation. So when we talk about computation, I'm not talking about how well you're able to use excuse me, how well you're able to use the computer. The thing that we are talking about is how well you know how to code. And is there one coding language in which you have at least some intermediate level of expertise? Can you write your own code, or can you interpret someone else's code from your understanding? So, computational biology has now there is not even one particular research that doesn't use bioinformatics. Almost there is some part of computation involved, but you have many different areas that require computation, and you can apply the data science techniques, the analytics into biological data. So bioinformatics, it's made up of two terms, biology and informatics. So basically, you use biological data with informatics or information technology to understand the biology behind it. And in computational biology, people interchangeably use these two terms. Uh, but computational biology is more about method development, algorithm design, and creating a generalized pipeline that a biologist can use for analyzing their data. So this is in a very nutshell of what bioinformatics computational biology is. And fortunately, CBSE has in many schools in the 12th standard has an elective course on bioinformatics. And so if it doesn't matter if they have taken up that elective or not, still if someone in an undergraduate handles biological data using algorithms and methods that are designed for taking up that data, then to some extent that is bioinformatics and computation.
Yeah. So that was a really wonderful explanation, and I still remember that I was into biology. So in 12th standard, we had all those terms and everything coming up, and then when we eventually move ahead in our careers, so such explanations are the pioneers of our career. So
Yeah.
that's really inspiring. So building on that, and I'd love to explore another aspect of your work. Your work spans protein engineering, antimicrobial resistance, genomics, and AI driven biology. What are some of the exciting questions your lab is trying to answer today?
The exciting question, the first and foremost, is this fun? Is this really something that excites me? And uh, how does it benefit science in the long run? So basically, the question is very broad, but the curiosity behind it that remains the same, irrespective of whatever area that we are working. So I have students who are working on network science. I have students who are working on OCR techniques or optical character recognition. I have students who are working on mathematical modeling, and someone who's using integrative computational and uh, biological data that's available in literature, and to come up with a model that can explain the macromolecular complex. So it's pretty diverse in that range. And what excites in each of these projects is that how does this work? Why does it work like this? Why is it reported like this? What makes this an exciting area, and what is the benefit of study? So that is what starts. That's the first level question you ask. Then, of course, we go on to more narrower hypothesis building, where we ask, okay, why does this particular organism need this macromolecular complex to degrade a polymer when that particular polymer is not the main carbon source for it to live? Then what is the use of? What is the evolutionary pressure that has gone into it? So you can see that the next level of questions that we ask is more biology. So biological perspective is very important in every uh, project, and students mostly who do not have biology background in my lab, we try to first make sure that they understand the biology behind it, at least to a level that they can decipher the data and also to understand the results that come on. So the questions, specific questions will change project to project, but overall theme is that the question why, and why is this interesting? Why does biology begin in this way? Why do we observe these things in this particular manner, and what's happened? So basically, the curiosity is what is the underlying point in all these projects.
Already there was a really great explanation, because I think that gives us such a wonderful foundation to understand your research is more and more deeply performed. Yeah.
Yeah, because you don't start anything with a
Yeah.
very specific question. If
Yeah.
you start with a very specific question, that means
Yeah.
you lost the forest for the woods. You
Yeah.
have to you have to start broad and then keep narrowing it down to a specific question that you want to
All
ask.
right.
Yeah.
Yeah, yeah. Thank you so much for the explanation. And heading to the next question, biology is becoming increasingly data driven. So, do you think future biologists need to learn coding? If yes, where should students start without feeling overwhelmed?
That's a good question. We are living in a big data world. Our mobile phones, our internet, whatever data you're buying, even apps that you're using, everything is tracking some kind of a pattern. And that pattern comes from the data the user is giving. So when it comes to biological data, we are soon and we are already in many of the instances in personalized medicine. And personalized medicine needs large amount of data, a sample size of one only from that patient, but a longitudinal, meaning that over a period of years. That means the question of whether this particular person will encounter a chronic kidney disease or will have a diabetes or will have some kind of health concern, how does that particular health burden can be mitigated? And to answer that simple question, which is very simple for humans, but a very complex question when you take from a machine or from a computer computation perspective. For it to answer, you need lots and lots of data points, and the lots and lots of data points come from a longitudinal data, time dependent data. So basically, to tell you if a person needs to be uh, admitted in an hospital for because of the previous incidents in having kidney disease, what is the a number of days that they will be inpatient? Now, this is a very simple question to ask from a economic, from a management perspective for a hospital. But for this question to answer, long couple of years ago, Google used three hospital data and they used electronic health records, and the number of data points ran in billions, and they were able to do the prediction with the accuracy of plus or minus 1 day, which means either they will be inpatient for 2 days or 3 days. So that was the accuracy that they got, but the number crunching that happened was using a huge amount of data. So escaping the data is unavoidable. You will be faced with a deluge of data. What type of data it is, how the quality of data it is, that is going to determine. And for determining and making sure that the data is of high quality so that the signal to noise ratio is very high, the most easiest way to do it is by cleaning the data. And data cleaning is semi-manual at this moment for many areas. So even if you take genome, even if you take proteome, even if you take agricultural data, it's very noisy. And to remove the noise, there is a human that needs to be present who looks at it and then says, this is a noisy data, this is a not a good data, or this is the only part that can be used. So coding becomes very important and essential in those cases. And if someone has not learned coding so far, and if they want to learn coding, the easiest one that they can pick up, and which is also used in industry, is Python. So they can start with Python. Yeah, sorry.
Yeah, so that was a really great explanation. I think now, so that we have spoken about your journey, let's shift our focus to the exciting science you are working on today. So, you have collaborated with researchers from different disciplines. From your experience, what makes a successful collaboration between computational scientist and experimental biologist?
That's a very good question because most of the time, like I said, there's an inside academic joke that computational people, all they need is a laptop and an internet, and they can just publish papers. It doesn't work that way. Not in 99% of the cases. So when experimentalists come to us, the first and foremost that we ask is, have you designed the experiment? Have you conducted the experiment already? If they say no, then that is where our role comes more prominent. We can help them to design the type of experiment that they want. Not that they don't know. It just helps us to think about data that they can give us rather than going back and then redoing something else. So a successful collaboration is where I have had where the experimentalist or the wet lab person has come to us very early on and then said, okay, we are thinking about this, we want to do something, and then we are thinking in these lines. What are your thoughts? And then when we give our inputs about how what type of experiments can be done, what type of data can be used, and when we design the computational experiments also, just like an experiment list, we tell them at what level of resolution the data tells us the evidence, or how much it supports the hypothesis. And to do that, the successful collaboration that I had is where we are frequently in touch with them. They are frequently in touch with us with weekly meetings, one-on-one phone calls, and then telling us what type of new results that they got, and so on. And so uh, it's like a two-way street, and it is kind of like a relationship where you have to work on. You cannot presume certain things that the wet lab person will do the experiment and give the data to. They might be encountering some issues that they will think it's kind of like trivial from the perspective of computational biology. But having a discussion and an open chat helps them to think differently, because we think from a different angle and they think from a different angle. And having been trained as a biologist, I, to some extent, bridge the gap between the experimentalist and computational person. So I can look at the experiment that they're doing. I can say them, okay, can we have one more control that can test for this? Can you repeat this in like in more triplicates than you are doing in duplicates? Do you have enough material for that to do? Because you encounter a lot of practical issues, you encounter a lot of economical issues, and so sociological issues to be very important. And so having a discussion with all the stakeholders end up having a lot more discussion and being a very productive collaboration. So all the collaborations that I've had, it's not like a one-way street where they give the data and they say, "Oh, do the magic." That rarely works, because I don't even know where the data came from, what did they do. So I need to know a lot more what goes in their mind. It's like a detective work, and also I try to be as transparent from my perspective to them, because for an experimentalist, they are not bothered about like what type of code I'm writing, or how many data points I'm using. What they might be worried about is they have a deadline and whether I'm able to meet the deadline and do the results. So that's when I try tell them, okay, this is a strategy that I'm going to follow. This is what I'm going to do. These are the computational controls that I'm going to have. Do you have any input that you can give? Because that will help in redesigning the computational pipeline as well. And so it's a two-way street. And those two-way street collaborations have been very, very successful in my experience.
I see. So that was a really, really, deep explanation. And I think many students listening here will relate to what you just shared. So, which leads perfectly to my next question. Um, Research rarely goes exactly as planned. Could you share a challenge or a failure that taught you an important lesson and changed the way you approach science?
Yeah. So if you, so there is this is not my idea, but there is a person who started a CV of failures. So basically, instead of a CV of, so usually your CV is a reflection of all the success that you got, the awards, the prizes, and everything that has been really. So they started a CV of failures where all the places that they applied for, but they never got shortlisted, all the awards that they didn't win, prizes they didn't win, and so on. So rather than a professional uh, my timeline, you can say that. you encounter failures in science more often than you would think. So every day you don't think about which experiment or which results will be interesting. Rather than you think about whether the script ran completely, did it have any issues, or there's a bug that I missed, and how do I troubleshoot it? So you go with an expectation that there will be some kind of a failure, some kind of a um, um, uh, some some miscalculation that could have happened, and so on. So you have to first of all like become very thick-skinned when you see failures rather than getting dejected about, oh my god, this code did not work today, or there was a power outage, my jobs got killed, and so on. Rather, take it up like, okay, fine, like it happened, now there's nothing I can do about it. Let me rethink it in a different way, and then can I come up with a plan B if plan A fails? So you always think about challenges in every project. So when it was the project that I did along with Karishma with the BN, we ended up having plan A, B, C. Like what happens if these antimicrobial peptides Sorry. These antimicrobial peptides are not having the enough data to be included in the database. And what do we do about them? Do we not consider them? Do we consider them? So ifs and buts. There are a lot of things that you have to think about. So every project, like even now, one of the students is facing a lot of issues with respect to the mathematical model that she's working on. She is not having all the variables that are required to build a model. And that frequently happens because we are the user of the variable, the data that has been given to us. We don't create the data. So what was there in the literature and in the public domain, we take it up and then we use it or we create a model with all those limitations. So that is one work that's happening, and she has probably failed in trying many different things, and so every day is a challenge. So there are multiple specific challenges that I can talk about, but that will take up more time. But what I'm trying to say overall is that challenges is something that you have to normalize. You will face challenges. You will face some setback. But what has to be is that, okay, it happened. That's fine. It's normal to have something that didn't work as planned. But what happens to plan B? What happens to plan C or D? So you should think about different scenarios when plans won't work out.
So that's truly enlightening, and I think that that really highlights how um, how interdisciplinary modern approaches has become, and particularly modern science has become. So, heading to the next question, artificial intelligence is transforming biology at an incredible pace. Which developments excites you the most, and where do you think human creativity and scientific intuition will always remain essential?
Yeah, that's a good question because we are right now living in the age of AI. Everything is AI, and AI has become the magic word for everything and anything. Um, we have to remember that AI is one another tool that helps us in getting the science done. So rather than having a binary outlook where in one extreme you are like completely trusting whatever the AI is giving, and the other extreme where you do not like use AI, do not like using any of the AI tools and so on. So there should be a balance. So use AI wherever it helps you to speed up the process. Like for example, you have a good understanding about the language, but then you don't want to reinvent the wheel and then write more things about the trivial coding part. Get it done with AI. And the vibe coding that's happening these days, it's unescapable. Everyone is doing vibe coding. But what we need to do is when you do a vibe coding, you have to make sure that it has all the loopholes and the flags checked. So when we do programming, we usually look for good coding practices. Did it also do good coding practices? Did it take into consideration the different scenarios of the user input that can go into making uh, um, the code work in all scenarios? So uh, the as I said, we cannot live without AI anymore. AI is going to be there. The human creativity and human reproducibility aspect is something that the AI is probably not going to replace. And I strongly believe at some point of time, we will have humans who are going to supervise the AIs and monitor what it's doing. We will, of course, in down the 10 years, we will have agentic systems which can automate everything, right from the performing a hypothesis, right from doing the experiment computationally, getting the results. And we already see it with Claude and the most recent models of the AI tools that are there, but it's going to be more prevalent. But what needs to be understood is that from a science perspective, is that we need to cross check, we need to make sure that the AI is just another tool. And also from the user perspective, we should make sure that there is an amount of cross-checking that happens when whatever AI throws at you. So that is something that I think every computational biologist or every person who's working in AI um, knows about it. And for newcomers, use it as one another tool that you can master and one another tool that you can um, get more things done in a very short period of time.
I see. So that's incredible. And speaking of the future of science, that's one topic that's impossible to ignore today. So that is artificial intelligence. Um, Moving to the next question, for students who want to pursue higher studies abroad or in leading research institutes, what skills and experiences make an application stand out beyond grades?
Okay, so we are a nation that's obsessed with marks and grades. But let me tell you one frank thing, that marks help you only up to the next level. So your 10th marks help you with your 12th, your 12th marks help you to get your undergraduate, your undergraduate marks help you only up to the next level. Your 10th marks are not going to help you in your PhD. Your 10th marks are not going to help you in your undergraduate. There is no correlation. And after some point of time, especially after the undergraduate, marks or grades don't matter. What matters is that whether, if let us say a student, uh, if they are going to industry, that's fine. But if they are going to academia, what academics look at is that whether this person has the ability to think, have the curiosity which they had as a kid, did not leave them after living in a very cynical world around them, where almost all the people around them keep saying, तुम बहुत ज्यादा बोलते हो, तुम बहुत सवाल पूछते हो, and those kind of things. Those are the things that kill curiosity in all of us. So, uh, if, in a marks and grade obsessed world, what you need to do or what one can do is to have a very high level of curiosity. Why does this work? What is this? What is this happening? Now, if you want to apply to someone's lab or anyone's work, you just reach out to them. These days, writing a cold email is not uncommon. It was a very rare event in our days. But then you can write a cold email. Many people do respond back. Many institutions do have open house where they welcome students to visit their lab. So look out for those events. The only thing is a lack of awareness about when does that happen and who conducts it. But when that happens, do ask questions. It can be any question. Like, for example, why are you working on a mouse brain? Why are you not working on a human brain? So that could be a good question to ask a neuroscientist. You can also ask why you are working on a operating system that I have not seen before. So I would say at that point of time that this operating system is Linux, which is what is more easiest for mathematics and computational values to train with rather than Windows. It's not that you cannot do mathematics in Windows. But compared to Windows, Linux and Mac or any other operating system that interfaces with the high performance computer is most easiest to do. So every question is a very good question to ask, and the most easiest way for a person to get into science, irrespective of whatever grades they have, is read a lot. So reading is something that they need to do, read biographies of scientists, read what challenges they faced and so on. And also one of the major skills that the student has to develop before joining anywhere for masters or PhD is communication skills. How to write proper grammatically correct sentences and how to talk in front of anyone, how to put across what you are thinking in a very easy way and not in a very confused way. And I'm not talking about accent or anything. I'm just talking like very simple English. So communication is one skill that we have learned in our schools. Most of us are English medium trained students who are very good at writing English. But when we say, okay, can you write something on your own about what you saw like the day before? They have some difficulty articulating their thoughts and views in a proper way. So reading, writing is something that is being slowly getting degraded with AI world these days. They have to do a lot. They have to have the curiosity and also uh, be very genuine in the question you ask to someone. So you cannot, because it's very easy to look through a BS that anyone talks about. So you can actually find out if a student is just regurgitating something that they just learned 2 days ago in school, or they understood it but have a very uh, basic genuine question that they didn't find answer from anyone. So those type of things are something that excites us as well, because when we look at students who want to join an institute or a lab, we see how good they will fit in our particular lab or an institution. And the way we look at it is whether it's not they learn this technique, whether they know this particular science behind it. That is not what we are looking at. We are looking at whether they have the ability to ask a genuine, honest, basic question. If they are able to articulate that in a very simple term, then they can understand abstract, complex ideas and convert them into very simple questions, which will help to generate hypotheses, experiment design, and so on.
Already, so that was really wonderful. So I think beyond the science itself, I would also like to talk about the people who aspire you to into this field, right? And looking ahead, what do you think will be the next big breakthrough in computational biology over the next 10 years, and how can students today prepare to be a part of it?
That's a good question. 8 years ago, the big thing, or 10 years ago, the big thing was big data. Data values, data information that's coming out, and so on. The cost at which data was generated was getting cheaper and cheaper, and now we have too much data. We are living in a like a surrounded by data. Now we are getting used to that, and now we are dealing with the issue of how to integrate different sources of data, homogenize it from heterologous sources, and uh, identify the patterns. That is something that we are dealing with, and we have been fairly successful. We, in the sense, not just my lab, but many other labs, everyone, we are looking at many new techniques that are coming. 10 years down the line, what would be more something that will come up will definitely have an AI component in it. And and of course, the rate at which the data generated will be much higher and faster compared to what we have. What we are doing today with the integration of heterologous source of data into a homogenized one, that might seem very easy to do 10 years down the line. And we might have much better techniques at a much better resolution of uh, high throughput. So that is something that we have achieved in certain areas, but not so much in other areas. So high throughput data driven, AI driven understanding is something that I think will be the next biggest breakthrough. And that will happen when we have the data integration pipeline on all these hiccups being sorted out, which we are facing today.
So that is incredibly amazing. So as we begin wrapping up our conversation, and so that was a really thoughtful explanation, and I would really love to hear your thoughts on what lies ahead of computational biology. So now we would love to hear from our audience. So this is your time to go. Please go ahead and ask your questions, audience. Ninath, can we have questions from the audience? Okay, okay, I see. So, yeah, so then, sir, would you like to go ahead with one last question or?
Sure, yeah.
Okay,
Go
all
ahead.
right. Yeah, so coming to the last question, finally, if you could give just one piece of advice to students who dream of becoming scientists but often struggle with self doubt or setbacks, or what it would be.
Usually, giving advice is not advisable because there is a the saying in Tamil, the food for today and the advice given by someone is only helpful for one day. Okay, so it's huge advices are not advisable, but I would say not as an advice, but as suggestions that the students uh, should be very curious, stay curious, be curious about many things. Ask the question. The most important question in science is why. So ask the question, why is this? Why is that? The person whom you are asking, they may not have the answers, but now everything, the answers are there. It may be a wrong answer, but at least there is some answer that you can cross check and verify and then see if that is the real answer. And uh, the, and the curiosity level, keeping it high helps you to achieve other things also. Like for example, reading from books, reading from others' biographies, reading from textbooks and so on. So why is this like that? Why is that like that? So that is something that I probably would suggest the students to have. And of course, in our constitution, the only country probably has it in our constitution that citizens of India to have a scientific temper. So this is again an ambiguous word which can mean many different things. But one aspect of scientific temper or keeping up the scientific temper is to remain very curious, and that helps us to have a very scientific approach to life and having a very scientific approach to looking at things.
Yeah, so that was really amazing. And I think before we would conclude, I would like to end that I believe every aspiring scientist would really appreciate hearing your thoughts on. And I think I would again give you a vote of thanks, because this was a really wonderful session that I am sure that all the audience would love to hear and implement the same into their particular journey of science and research. So, Dr. Ragothaman M. Yennamalli, thank you for taking the time to share your inspiring journey, valuable insights into computational biology, and your vision for the future of science with us today. Your words have not only deepened our understanding of data driven biology, but have also motivated aspiring researchers to embrace curiosity, interdisciplinary learning, and innovation. It has been truly an honor to have you with us at the next experiment by the SciComm Science. And to all the lovely audience, a sincere thank you for having your enthusiasm on the podcast and participating, and your thought-provoking questions made today's conversation even more engaging. And it's been a pleasure hosting you. We would really appreciate if you all can take a moment to really share your feedback for our next event to make it better and energized one. There's a link of the feedback form in the comments to receive e-certificates. Please like, share, and subscribe our YouTube channel. And for more information, do visit www.thescicommsynapse.in. And until then, think, share, and grow. Thank you
Thank
so
you
much.
so much.
Yeah, thank you so much.
Thank you.
Have a
Thank
great
you for SciComm and the next experiment. Thank you for this opportunity. I really enjoyed talking
day.
to you, Dharani, and Minaj. Thank you for that. Thank you.
Yeah, yeah, it's been a pleasure to have you on stage, and taking all the knowledge that you have, because I believe each and every scientist and researcher is actually having ocean of knowledge that we cannot pick up in an hour or so. It takes a lot of years to understand that.
Mhm. You have to remember what we know is just a size of a mustard seed.
Okay.
What is there is a size of the world. So our knowledge is only limited.
All right, thank you so much, sir.
Thank you so much. Have a
Yeah,
great day.
yeah,
Bye.
same to you. Great day. Bye, sir.